I work at the trade school on the north side of the Charles. I've been there for a very long time. During that period of time, I've become a big chauvinist for the city of Boston, not having been born here. Believe it or not, we have the greatest concentration within a radius of a mile or two, the greatest concentration of biomedical researchers in the world, right here. It's really quite extraordinary. I'm not saying this purely out of Boston chauvinism, but also for reasons of demography. I also noted, and I thought this was politically incorrect to say so, but then Kelly Tuthill said it, that the assembled congregation today is unusually elegant and attractive. And I don't mean, again, to rub salt in the wounds of the New Yorkers, but I think... But I digress.
No offense taken.
Whatever. So I work on how, when I say I mean the people in my research group, on how cancer cells invade and metastasize. Because as you likely know, virtually all of breast cancer-associated mortality doesn't come from the primary tumors, it comes from the distant growths when the cancer has spread. And we haven't really understood until the last 10 years, and now we begin to understand it in great depth, what causes cancer cells to leave the primary tumor and take up residence in distant tissues where they become life-threatening, the process of metastasis. In fact, cancer cells in primary tumors don't naturally invade and metastasize. They have to learn how to do so. And when they learn how to do so, they reawaken early embryonic behavioral programs that have long lain silent within them. And when they reawaken these programs, they can then exploit them to propel them outside the primary tumor through the circulation and to distant tissues. We also know that breast cancer can be divided into different subcategories, luminal and basal A and luminal B and so forth. What is not so obvious to many people is that within a given breast cancer, within a given tumor, there are different gradations, different subpopulations of cancer cells. Within a given tumor, there are, for example, good guys who don't spread, and bad guys. The bad guys we often call cancer stem cells. The good guys are the non-stem cells within the same tumor. They cohabit the same tumor. And the question is, what controls the proportions of the bad guys and the good guys? Over the last couple of weeks, we actually found something pretty interesting in my laboratory, and we're now collaborating with Dr. Andrea Richardson here from the Brigham and Women's Hospital, who's here, to validate this, because we don't work on people. I'm not a real doctor, but she and they over there are. And it's the following. If you look at a more benign luminal breast cancer and you ask, how often do the good guys become bad guys? The fact of the matter is they virtually never do so. They somehow are constrained from becoming cancer stem cells, and that is clearly part of the reason why they have such a much better prognosis. Conversely, as we discovered in the basal cancers, if we look at the good guys in the basal cancers, or we thought they were the good guys, they have a great proclivity, a great tendency to flip over and become bad guys. And that is to say, to become cancer stem cells. And we now have traced that to minute molecular defects in the chromosomes of those cells. In telling you this, I'm saying that we begin to understand now in minute detail why and how breast cancer cells behave, what governs their behavior. This is not immediately translatable into clinical therapy, but people like me work with the faith that if we understand the molecular underpinnings that lie deep in the heart of individual cancer cells, we will then be educated and informed as to how people like Judy Garber and others can actually develop ways of treating the disease. And conversely, if we don't understand what makes a breast cancer cell tick, we have no hope of developing new ways of treating this disease.
Thanks very much.